Good morning, students. Today, let's learn about Mendel's monohybrid cross. In this monohybrid cross, we learn about inheritance of one trait, and this is going to involve a single gene, and that is why the term monohybrid cross. So, what Mendel had done was he had taken a tall plant and he crossed that with a dwarf plant. Now, when he crossed a tall and dwarf plant, when he got the F1 progeny, all the plant turned out to be tall. And there was nothing that was showing the dwarf trait. Now, he went on further to experiment and he did something called self-crossing or known as selfing. And when he self-crossed these tall plants from this particular generation, he found two different outcomes. So there were tall plants and the dwarf plants that were seemed to be missing in the previous generation reappeared here. And of the total outcome, 3/4 were tall plants and 1/4 were dwarf plant. So in other words, we can say that there was 75% of the entire progeny or the entire offsprings that were having this tall trait, while only the 25% that was showing the dwarf nature. So let us try to understand the genetic basis of these cross and these outcomes. So when we talk about the tall plant, right, we will, let's say, we start with the parent, how we solve monohybrid cross. We start with the parent, and here the parent as per Mendel were the tall and dwarf plants. Now, tall plants are going to be regulated by one type of allele, and the allele is going to be the capital T. So the capital T allele is going to regulate the tall trait, and the small t regulates the dwarf trait. Okay, now when we talk about again the tall plant, these are going to be diploid in nature. That is, they're going to have a paired allele structure. So here they're going to have the same type of allele, right? And that is going to be capital T, capital T. And here they are going to have the small t allele. Now, another thing that you have to keep in mind at this point is when you're going to have the same type of allele in a diploid condition, they are called homozygous or homozygous nature, we can call it. So small t or capital T is called homozygous. Now, obviously, when they are going to undergo the process of sexual reproduction, they are going to produce the gametes, and the gametes are going to be haploid. So haploid means they're going to have only one set of chromosome. And when we are talking about one set of chromosome, there is going to be only one allele that is going to be present in the gamete. So each of the gamete that is going to be formed is going to contain either that of the capital T allele or the small t allele depending on the parents. So in this case, this tall plant is going to produce only one type of gamete that is going to contain the capital T allele. It's capital T and capital T because both of the gametes are going to have the same kind of allele. We are going to write only one. And here on this side, we are going to have all the gametes with small t alleles. Now, when they are going to fuse, they are going to produce the F1 progeny. So they will undergo the process of fertilization, right? Pollination and followed by fertilization. And when they do, they will form something called the F1 progeny or F1 offspring. These are nothing but the hybrids. And again, they will be diploid in nature because we are fusing haploid gametes, two haploid gametes to get a single diploid structure. And here the progeny that Mendel found, as we have already talked, were all tall. Okay, and the allelic composition in this case is going to be capital T, small t. Okay, so this capital T, small t condition, right? This is going to be known as heterozygous. Okay, so when you're going to have the same type of allele, it is called homozygous, and when you're going to have two two different type of allele, it is going to be called heterozygous. So we have got a heterozygous condition, and this heterozygous condition, we are having the expression of only one of the parental trait, that is the plants are all tall, and we are seeing nothing that is going to be dwarf. The question is whether the dwarf traits are lost, but again we have already seen that they are actually not lost, they are still present, but they are simply not expressed. So from here on we have to go to the next generation, right? And this next generation is called F2 generation, and first we are going to start with F2 parents. Okay, and in the F2 parents, what Mendel did, he took the F1 progeny, right? He took the F1 progeny only and he crossed them. So he took the tall plants, okay? He took two F1 progeny, he took the tall plants with the genotype capital T small t, capital T small t, and he actually self-crossed them. That is called selfing that we are talking about. Now here also there is going to be the gamete formation just like the previous case, and as we had said that during gamete formation the alleles here will segregate in such a way that the alleles will be present in only one in each of the gamete. That is, each of the gamete is going to contain only one type of allele. So based on that, the gametes can contain capital T allele or small t allele. Similarly, on this side also it can contain either capital T or small t allele, but never both. Now we are talking about F1, F2, right? So this F1, F2, what do they represent? So here you have to remember that the F1 represents the first filial generation, okay, and the F2 represents the second filial generation. So moving on, once we have the gametes here, right, for this F2 generation, we have to find out the outcomes, we have to find out the progeny here. So we have to talk about the F2 progeny, and again to find out the F2 progeny we need to do something called, we need to make the Punnett square, also known as the checkers board. Okay, all right, here you see the Punnett square. Now in the Punnett square, what we're going to do is we're going to represent the gametes in these four outer boxes, and here we will let's say if this was the male parent and this was the female parent, so let's say we are going to have the female gametes here and the male gametes here. So accordingly, the gametes that we had got for the female, let's say it is going to be capital T and small t, and for the male here again with the capital T and small t. Now when we are having these gametes, they will obviously fuse during the process of fertilization and they will give us the outcomes. So here capital T, here is going to be capital T and small t, so capital T and small t here. So here also small t and capital T, so we can always write capital T and small t, and in this one you're going to have the small t and small t, we are going to have small t and small t. So when you're going to have this particular allelic composition, right, what is the visible expression, right? This is what is a condition at the genetic level, allelic level. So what is the expression of this actually? Now the expression that we're going to talk about in the first case is going to be tall plant, in the second case tall plant, in the third case tall plant, and in the fourth case it is going to be a dwarf plant. In other words, what we are seeing is when you're going to have a heterozygous condition, as we're talking about capital T and small t, we are seeing the expression of the parent that had capital T, capital D. That is, in other words, we can say that the capital T that was regulating the tall trait is actually being expressed even in a heterozygous condition. And for the homozygous condition, homozygous trait to be expressed, it is always requiring the homozygous alleles to be present. Otherwise, you can see in none of the other condition, right, when we are going to have the homozygous condition or heterozygous condition of the capital T alleles, we are going to see none of them having the expression of the tallness, but only when we are going to have the small t, right, homozygous, only then we are going to have the dwarf trait. Anyway, so from here we are going to talk about something called dominant allele and recessive allele. As you can see, even in the previous case, what you see here in the F1 progeny, you see the expression of the tall trait and the dwarf trait is missing. So here we had said that the capital T was actually regulating the tall trait and small t was regulating the dwarf trait. So even in the heterozygous condition, what you're going to see is the expression of one of the trait that is dominant over the other. So the trait that is being expressed even in the alternate or even in a heterozygous condition over the alternate allele, one allele is going to express themselves and that allele is going to be called the dominant. Okay, it is called dominant allele. And what is the dominant allele for us? The dominant allele is going to be capital T. And the second one is going to be the recessive allele. Now the alternate allele, right, or the allele that is not able to express themselves in a heterozygous condition, that is going to be called the recessive allele, and in this case it is going to be the small t. So typically what we do, we represent the dominant allele in a capital letter and recessive allele in a small letter. So remember, only the recessive trait is going to be expressed when we are going to have homozygous recessive condition. So if I go back here, now we can understand that here we can say the capital T be to be homozygous dominant. Okay, homozygous dominant, and the small t to be homozygous recessive. Okay, homozygous dominant, homozygous recessive, and then we are going to have the heterozygous condition. So these three, right, these three allelic composition is referred to as the genotype. Okay, it is referred to as the genotype of the organism for that particular trait, and the genotype is going to regulate the visible expression of the trait, and that visible expression of the trait is going to be referred to as the phenotype. Okay, it is going to be referred to as the phenotype. So phenotype refers to the visible expression of the genotype. So genotype is the allelic composition and phenotype is what we are able to see. So capital T is going to be dominant allele and small t is going to be recessive allele. And again, genotype can be of three different types at any particular case when we are talking about a monohybrid cross. So from this F2 progeny, right, we can count the number of plants that is showing a particular type of trait, right, the phenotype we can say. So what are the different phenotypes we are going to have? We are going to have two different phenotypes, that is tall and dwarf. And if you are going to count that, here we are going to have tall 1, 2 and 3. So there are going to be 3 tall and only one that is going to be dwarf. So when we talk about phenotype and phenotypic ratio, right, we are going to have a ratio of 3:1 for the tall is to dwarf. Now what about the genotype? So we are going to have how many type of genotype? This is one type. So capital T is one type of genotype, that is going to be homozygous dominant genotype. Then we are going to have the heterozygous genotype, that is capital T small t, and finally we are going to have the homozygous recessive genotype. And you have already seen that it is going to be of 1 for the homozygous dominant, 2 for the heterozygous, and 1 for the homozygous recessive, as you can see in this particular case. So this one is referred to as the genotypic ratio or genotype ratio. And for a monohybrid cross, the genotype ratio is going to be 1:2:1 always if they are going to follow the Mendelian inheritance. So the students, you have to understand two ratios here in monohybrid cross, that is going to be phenotypic ratio for the tall is to dwarf, that is dominant to the recessive trait in the ratio of 3:1, and genotypic ratio in the form of 1:2:1 for the homozygous dominant, heterozygous and homozygous recessive. So what were the laws, what are the conclusions, generalization of the laws of inheritance that Mendel had proposed from this? So there were two of that. So these two were the law of segregation, I'll tell you what these are, and second was the law of dominance, as we are talking about. So what the law of segregation states is, during gamete formation, so when we are going to have the gamete formation, there is going to be segregation of the alleles, and this allele, this paired allele, segregates or separates in such a way that each of the gametes that is going to be formed, they're going to contain only one of the paired allelic structure and never both, and that's the law of segregation. And remember, this is going to be universal. In any case, the law of segregation will hold good, because during gamete formation the alleles will have to segregate, and that is what you have to understand with the law of segregation. And you can explain this with the help of the monohybrid cross. For example, even you can see in the case of F1, right, or even in the case of F2, the gamete formation, segregation takes place, and each of the gamete is containing only one of the paired allelic structure. Now, what about the law of dominance? So, the law of dominance talk about the dominant and the recessive allele. So, it says that in a heterozygous condition, see, we're talking about heterozygous condition, there are two different type of alleles, and one of the allele is going to express itself even in the presence of the alternate allele. So, of the two allele, one is able to express, and that allele is called the dominant allele, and the other allele that is not able to express, that is simply present, and that is called the recessive allele. And we already have talked about the dominant and recessive allele. So the example that we had taken of the stem height in this case, and in this case we had taken the two different alleles, capital T and small t, and this capital T allele was a tall trait, was able to control the tall trait, and the recessive allele, that is the small t, was able to regulate the dwarf trait. So each of the allele is going to regulate one trait, and typically there is going to be a dominant allele and a recessive allele. So here are the things. If the plant is going to be having a homozygous dominant genotype, as you can see in this case, then obviously the tall trait is going to be expressed. When you're going to have the homozygous recessive, the recessive trait is going to be expressed. But when we are going to have the heterozygous genotype, that is capital T and small t, in those cases it is the dominant allele, it is the dominant trait that is going to be expressed, and that is what precisely the law of dominance. Thank you all for watching.